Hi, everybody. This is Dr. Jonathan Kuttner. I want to tell you an amazing story, and the story comes from the files from the CDC, the Central Center for Disease Control in the States. It's like a mystery novel. It's almost like an Agatha Christie mystery with twists and turns, and it's utterly fascinating. And it kind of forms the background for a lot of the other discussions that occur around the coronavirus. We're living in the middle of this enormous pandemic. And a lot of the experts stated unequivocally beforehand that it's not if, it is when the next pandemic's going to come, and here it is, we're living in it. The same experts have been saying that once this pandemic has been playing its way out, where the coronavirus has a fatality rate of maybe 3 or 3.5%, that this may well be just like a teaser for the real deal, for the real pandemic, which is, and it's again, not when, not if, sorry, it's not if, it's when a worse pandemic is going to come. And they base this on the fact that previous pandemics, we've just been super lucky. So if you look back at previous coronavirus pandemics, which the one was SARS in 2003, where the mortality rate, the fatality rate was 10%. And then there was MERS, which came from camels from Saudi Arabia. The fatality rate there was 30%. A third of the people died. But the difference was that the virus didn't spread so much. So, and other pandemics have also, it's just we've been lucky up till now that we haven't had a massive pandemic until you go back over 100 years ago to the flu pandemic. And the flu pandemic was 1918. This was an influenza virus, one of many viruses, but the influenza virus is the one that the experts actually picked as the most likely. And the influenza virus of 1918 infected almost a third of the world's population and killed well over 50 million people. So now that is a pandemic with capital letters. And the real problem, once you hear this, you think, oh my God, so the next pandemic may well be something like that. Now, once you hear that, you think, oh, wouldn't it be interesting if we could, with present technology, go back in time, and if we could actually see what that virus was, the virus that killed 50 million people. And this is the story. This is the story that's in the CDC files. And so if we go back to the 1918 pandemic, go to a God-forsaken tiny little place on the west coast of Alaska, which is called Breivik Mission. There were 80 people there, mostly Inuit, and it is thought that some guy came on a sled. There was a trader, dropped in, maybe picked up some furs or did some swap with the people living there, 80 people living there, left, and then 5 days later, the flu exploded. This influenza virus exploded, and in 5 days, once the first person presented with the influenza symptoms of fever, rigor, cough, what happened was that over 5 days, 72 of the 80 people who lived on this tiny little place all died. And they all died of respiratory failure. Poof, only 8 people left. And these 8 devastated people must have made contact, and people from around came. They took all the bodies and they buried them in a mass grave covered with lots of little crosses. And because it was way up north in Alaska, the grave was encased in permafrost. So in 1951, Johan Halton, who was a Swedish microbiologist, and he was living and he was working at the time in Iowa in the States. For a postdoctorate thesis, he thought, well, wouldn't it be interesting if I could go up to, because he read about Breivik mission, and he thought, wow, well, all these people who are encased in frost. So if I went back, maybe I could take a sample of that from somebody and bring it back, and we could maybe use that sample and learn something about the actual virus that caused the 1918 pandemic. So he flew in a DC-3 there. He asked the people who were living there were more people there at the time, and the elders of the village gave him permission. He had to keep a fire burning constantly to kind of melt the ground, dug down, and then he found a 6-year-old girl, perfectly preserved, still wearing her blue dress and apparently red ribbons in her hair. He took lung samples from her and 3 other people, put them in a container which he had to keep cold, climbed back into the DC-3, and it's a prop plane, and it had to keep landing to get more fuel. And each time it landed, chaos ensued. And by the time he got back to Iowa, all those lung samples were completely ruined. And so he kind of sadly thought, oh, well, thus with life, and he went on with his life. So that's 1951. In 1997, now Johan Halton is now 72 years old, and he's paging through a microbiology journal and comes across an article written by a man by the name of Dr. Taubenberger, who's an American, who together with some other workers had taken samples, because this idea was there, tucked away. If we could only learn what that virus was that killed 50 million people in 1998, in 1918, maybe we could kind of prepare ourselves, learn to defend ourselves against something like it. So what Taubenberger had done, this was completely separately, is that he had found that an American 21-year-old soldier had died in 1918, and his lung had been preserved in a museum, in a medical museum. And he thought, oh, wow. So he went to the medical museum. He collected the samples of the person's lung, the soldier's lungs, and using, now we're in 1997, so using gene sequencing, he was able to sequence now the flu virus, just like coronavirus, has RNA in the middle. So, and there are 8 strands of RNA in the flu virus, and he was able to sequence 4 of the 8. So he had an incomplete story, and he put this article out saying, we have partially sequenced the genetic material in the virus that caused this incredible pandemic. And Halton, who was now 72, read it, rang him up and said, hey, and told him the story of how he went all the way up to the Breivik mission. And he said, can we do it again? And Tambo says, absolutely, go for it. So this time he paid for his own flight. This is Johan Halton, 72 years old. He apparently took his wife's garden shears with him to help with the excavation, which is very famously he took his shears. He flew over, apparently it cost him over $3,000. He landed in Breivik Mission. He again spoke to the elders and they said, yeah, guess so. So this time he and a few colleagues dug down into that same mass grave and they found a perfectly preserved 20-something-year-old Inuit lady who he named Lucy, don't know why, and he removed her lungs and took them, put them, now it wasn't sketchy, now it was very highly technical, he put them in exactly the correct preserving material, and they flew back to, I think it was Maryland, where they were doing this with Taubenberger. And what they successfully did was they took the samples and they sequenced all 8 segments of the RNA. So that's now how many? 70 years after finally they've got the virus and they've got the DNA of the virus. Now, having the DNA of the virus is useful. Having the RNA of the virus, having the genetic material is useful, but actually to really understand how the virus, why is this virus so lethal? And the very odd thing about the 1980 pandemic is that the age group of people who died the most, 15 to 35. Wow, terrible. Destroyed a generation of people. So what they then needed to do, and this decision was made very, very carefully, they had the actual RNA of the virus, wasn't alive, was dead, and what they wanted to do was now recreate the virus itself. Oh my God, recreate the virus itself so that they could then grow it in mice and other animals to understand why it was so virulent. And so they then used a new technology, and they used the services of probably one of the leading virologists in the world, a guy called Peter Pelisse, who is another American virologist. And here is so what they did was Pelisse took something called a plasmid. And a plasmid, when you have the virus, you have a spherical outer membrane. Then you have the virus particle itself, which is the RNA. Now, plasmids are something else that occurs in nature where you find floating in that virus or floating in the cells of the person that the virus has attacked or the animal that the virus is attacking. You find little circular bits of DNA. And the circular bits of DNA, if you break them open at the right place, they fit onto and take the shape of the viral RNA. So they create a template. And if you see plasmids around, what you know is this is one hell of a virulent organism, because the plasmid spreads. It's like taking the template and it's taking it away and spreading it further. So Pelisse very carefully took a plasmid shape of the RNA of the virus that came from that caused the 1918 pandemic. Then taking that plasmid, another virologist by the name of Dr. Tumpe went to a laboratory. Now, why this is important is that what he did was he recreated a live virus. So the laboratory was a level 4, which is the absolute top level, because he was recreating what was probably the most deadly virus, the sort of serial killer virus that we've had in the history of the world. The security was massive. Only he could do it. So for him to get in, he had to use his biometric thumbprint. Then, before he could open the cabinet where the virus samples were kept, they read his iris, which is apparently unique as well. He only came after hours when there was no one else working. The area that he worked, nobody could go into. He wore a suit which poured new air through it, and he used double gloves and everything, you know, that like the whole place was so supremely to be level 4. You know, the safety measures have to be absolutely perfect. They can't be less than that, because if it gets out. So working like this on his own, he took the plasmids that Palise had created. He then took those plasmids and he injected them into human kidney cells. So now you took the template for the RNA. Remember that the plasmid is what spreads the information of the virus. He took it and he put it into human kidney cells, which were just growing. They'd been grown on a special matrix. He put that inside, and what the plasmid did is it forced the human kidney cells to start making the flu virus. So it started production. Our cells are the ones that produce the virus particles. So that's what he created from the plasmid, which took the template of the 1980 flu virus. He put it into human cells. The human cells started making the virus. Now they had the virus itself, which is the virus itself, you know, flashing lights, the whole thing. And what they found on the flu virus, there are on the surface, you know, like the coronavirus, we see many, many pictures of coronavirus with little bumps coming out of it. The flu virus is very, very similar. It's RNA in the middle, and it has little bumps, but bumps of two kinds. And these bumps form the most, what is called antigenic. So these bumps are what your body recognizes as a foreign intruder. And there are two kinds of bumps or little spikes that come out of a flu virus. There are the H, or hemagglutinin, and the N, or neuraminidase. Now, the H bumps, the hemagglutinin bumps, what they do is they attach to your cells, and then they open up the cell and go in. So the H antigen, the H bump, is the one that is the degree of infectivity. The N bump is an enzyme, neuraminidase, which is, it cleaves the virus from the cell and allows it to get out and go. And so now there are 16 H and there are 7 N. So when you see a virus that says H4N5 or something like that, what they've done is they've worked out what the H antigen is and what the N antigen is, and therefore they say this is what the virus, this is the actual outer surface of the virus. It doesn't completely explain the unique qualities of the virus, but it tells you a lot about it. So H1N1, or whatever the number is, designates what the virus is, and it also tells you something else. It gives you a strong idea where the virus comes from, what's called the phylogenetics of the virus. So what they found was, so now once they'd made this flu, the actual virus, they brought it back to life. It's a bit like Jurassic Park, where you have a Tyrannosaurus cell sitting in some amber, and then you grow the Tyrannosaurus. So what these people have done is they took the tissue, and they now have grown, using human kidney cells, they have grown the actual virus itself. And they found out that the flu virus that caused the pandemic is an H1N1, and that the H component is actually a classical swine flu, and the N component is a bird flu. Ooh, okay, so that's where they came from. And the mixture of the actual the actual mix in the virus was unexpected. And it was unexpected because we've had a lot of pandemics. We've had one in 1968. We had another one in 1977, I think. And then we had the H1N1 swine flu in 2009. And there have been other which have been pandemics. Some have killed 1 million people. Now, what they've found, because they've been able to, they've kept these as isolates, and they're trying to work out what is it that actually makes a flu virus particularly virulent. And the pandemic virus had very few, if any, of the actual markers that makes a virus virulent, that makes it kill people. But when they studied it more, they found that it actually had, it had a unique mixture of 4 or 5 minor changes, which all put together explained why this virus, when they grew it, in places was from 100 to 30,000 times more lethal than the normal seasonal flu viruses. And these are minor changes, minor changes. Just that, they rolled the dice, and there it was. That's because that's what genetic mutation is. It's just a roll of the dice. So the last part of this marvelous article discussed lessons for the future. Now that we know, and this article was written way before this pandemic. It was sitting in the archives of the CDC. And they said, well, now that we understand, we've actually recreated, we've recreated this virus. We, under super careful, stringent protocol, we're learning so much more about what makes a virus deadly. And we understand that it's just the roll of the dice. So what we have to do, and that's why they say, it's not if, it's when the next pandemic is going to come. And they said, therefore, now what we can do is we can prepare. And they put in a whole, why am I laughing? It's quite sad. They put in a whole suite of things, setting up a pandemic commission, setting up, making sure you've got the facilities to make vast amounts of vaccine, making sure you've got the facilities to absolutely work out what the virus is, making sure you've got the facilities to just swing into motion and trace people who've got ill, trace everybody who they've been in contact with, contain, etc., etc. We know all this, and we know that this time round, too many countries had nothing or very little, or like America had a pandemic unit which was disbanded. So the lesson from this now is that, yes, we will get through this pandemic like we have all the others, but there are lessons to learn, and these lessons have to be learned. Thank you.